Hi, my name is Brooke, and today I'm going to be telling you about CRISPR-Cas9 technology. CRISPR is actually a natural process that is part of a bacterial immune system. In 2007, however, female scientists Jennifer Doudna and Emmanuelle Carpentier pioneered the revolutionary technology that allows scientists to edit, delete, or completely replace your genes. They even won a Nobel Chemistry Prize in 2020 for their work. But why would I need this CRISPR technology to fix my genes when I could just do it myself? No, not that type of gene. I'm talking about genotype, like your DNA. DNA is made up of 4 bases that precisely pair and string together to make the instruction manual to our bodies and determine our every trait. And like a real instruction manual, sometimes there can be mistakes. These mistakes in genes are called mutations. So what does CRISPR have to do with genes? CRISPR is a natural process that is used to prevent a virus's DNA from multiplying inside of a bacterium. Scientists have figured out how to change CRISPR to target DNA in almost any organism. Wow, that's really cool. How does it work? There are 2 main parts to the CRISPR-Cas9 system. A single guide RNA molecule made from clustered, regularly interspaced short palindromic repeats, or CRISPRs, and CRISPR-associated proteins, or Cas9. When these 2 come together, they form the Cas9 complex, and this is what is going to be actually going into the cell and performing the CRISPR process. Let's say that this person has a mutation in one of their genes which is causing them to have a peanut allergy. In the lab, scientists would design and create a guide RNA that matches the specific gene causing the allergy and attach it to the Cas9 protein. Once the Cas9 complex is inside of the cell, guide RNA directs the Cas9 protein to the targeted gene. After binding to the gene, the guide RNA will unwind part of the double helix and pair it to itself. Next, the Cas9 protein will act like a pair of molecular scissors and cut the DNA. What's cool about CRISPR is that there are a few different ways that it can affect the gene. Once the DNA has been cut, the cell will try to repair itself through a process called non-homologous end joining. This process leads to the equivalent of turning the gene off. That being said, back to our peanut example, if scientists add a separate sequence of template DNA to the Cas9 complex, cellular proteins will go through homology-directed repair, where after the DNA is cut, they use the template DNA as a blueprint of how to rebuild the gene so that it is not mutated, or even replace it with a different gene. Now that the gene causing the nut allergy has been replaced with a non-mutated gene, this person no longer has a peanut allergy. So if CRISPR can cut DNA and replace it with a different gene, does that mean that I could change my eye color, hair color, and even my height? Technically, yes. However, the power of CRISPR-Cas9 technology should be used to find new cures for genetic errors like hearing loss or cystic fibrosis, instead of for cosmetic reasons. Don't you think it would be better to reprogram drug-resistant cancer cells than to make your eyes purple? Yeah, I guess you're right. I'll just have to stick to colored contacts.